Let's begin today's class with TS of mature anther. TS is the transverse section of anther. You know the structure of stamen is stamen which possess anther and filament. This is the anther and it is filament. Now this stamen consists of anther and filament. The question is TS of mature anther, the transverse section, the section of mature anther. It shows the transverse section of mature anther reveals the presence of anther cavity surrounded by anther wall. In this anther there are two lobes are present, so it is called as bilobed. This anther is bilobed. Each lobe consists of two theca, that is dithecus. Dithecus. The bilobed anther which consists of two theca called dithecus anther. A typical anther is tetrasporangiate. When you take a section of this, it shows a tetrasporangiate. Tetrasporangiate. A typical anther structure is tetrasporangiate. Tetrasporangiate anther. So this is the diagram, the TS of mature anther, which reveals the presence of anther cavity. These are the anther cavity which is surrounded by the anther wall. Let's learn the anther wall. We discussed in the earlier class also. The anther wall, there are number of wall layers. They are epidermis. The outermost layer is known as epidermis. Just below the epidermis there is a layer endothecium. Inner to the epidermis there is a layer called endothecium. Then inner to endothecium there are 2 to 3 layers of middle layer. Middle layer and innermost layer is called as tapetum. Let's learn one by one the structure of this layers. Epidermis. Epidermis is the outermost layer and it is protective in function. Look at this diagram. This is the outermost layer is epidermis, which is made up of a single layer of cell. The function of epidermis is protection, which gives protection to the anther. Inner to this epidermis, this is the outermost layer, just inner to this epidermis there is a layer called endothecium. The layer of endothecium is elongated. The epidermis cell is made up of single layer, endothecium also made up of single layer of elongated cells, which contains the tangential walls of this endothecium consist of alpha cellulose. There is a thickening. Thickening is present in this endothecium. And this endothecium layer is, the nature of this layer is hygroscopic. Hygroscopic in nature. That means it's tend to observe moisture. That is hygroscopic. This endothecium is absent in certain plants, mainly in the aquatic plants. Aquatic plants and clistogamous flower, saprophytes, this endothecium layer is absent. So inner to the epidermis there is a single layer of cell which is called endothecium. The nature of endothecium is hygroscopic. The nature of endothecium is hygroscopic, which contains a bands of cells, alpha cells, bands of alpha cellulose cells are present in the tangential walls, sometimes in the radial walls of the endothecium layer. So, second layer of the anther. So, this is the structure of anther wall. The walls of anther. First layer is epidermis, second layer is endothecium. Inner to this endothecium. Now, the innermost layer, inner to the endothecium, there are 2 to 3 layers of middle layer. 2 to 3 layers. Now, inner to this endothecium, there are 2 to 3 layers. Either 2 layer or 3 layers of middle layer is present. Normally, this middle layer is ephemeral. Ephemeral, which is disintegrate very soon. So, this ephemeral layer, it's not permanent, which may be disintegrate very soon. That is called ephemeral. So, middle layer is ephemeral. This endothecium is hygroscopic, and middle layers there are 2 to 3 layers. Now, the innermost layer of this anther is called as tapetum. The innermost layer, this is the innermost layer called as tapetum. So, outermost layer, this layer is epidermis, next layer is endothecium, and this layer is middle layer, and this innermost layer is called as tapetum. This tapetum is innermost layer of the anther. So, this tapetum is dual in origin because it is derived from the peripheral wall layer, part of the tapetum derived from peripheral wall layer, and another part is derived from this connective tissue. This is the region called connective. Tapetum is dual in origin because it's derived from 2 different parts. Part of the tapetum derived from peripheral layer and another part derived from connective. So, it's dual in origin. And what's the function of tapetum? The tapetum, the well development of tapetum is seen at the stage of tetrasporangiate. When the microspore is formed earlier in the form of tetraspore. In this stage, tetrasporangiate stage, this tapetum develops well. The function is it nourishes the developing microspores. In the earlier forms, the microspores are developed. After tetraspore, there are 4 haploid nucleus is present. Each one derives as a microspore. So, this tapetum nourishes the developing microspores as well as the microspore mother cell. So, this is the function of tapetum. The function of tapetum is important. Tapetum nourishes the developing microspores. There are 2 different types of tapetum. They are named as secretory tapetum and invasive tapetum. There are 2 different types of tapetum: secretory tapetum, invasive tapetum. Another function of tapetum is it contributes sporopollenin through Ubish bodies. Sporopollenin. The function of tapetum, 2 more questions. What's the function of tapetum? Tapetum nourishes the developing microspore and the microspore mother cell, and it contributes sporopollenin through Ubish bodies. Sporopollenin is the structure which is present in the pollen grain, the outer layer of pollen grain, XII. XII is made up of cellulose and sporopollenin. So, this tapetum, it contributes the layer sporopollenin. That's the function of tapetum. So, tapetum is the innermost layer. Now, 4 wall layers are there: epidermis, endothecium, middle layer and tapetum. Now, there is another structure is found here, is called as connective. The connective is a column of sterile cells. So, there is a column of sterile cells are called as connective. Then microspores. Inner to this anther wall there are cavities. As I mentioned earlier, a typical anther is tetrasporangiate. It is tetrasporangiate. So, this sporangium, microsporogenesis. What is meant by microsporogenesis? Development of microspore from microspore mother cell. When the microspore mother cell undergo meiotic division, it forms microspore. Now, the microspore is it develops from the microspore tetrad. At first, the microspores are formed in the form of microspore tetrad. From the tetrad, microspore tetrad, it becomes microspores. This microspore is haploid. Microspore is the first cell of the male gametophyte. So, this microspore, how it derived? When the microspore mother cell undergo meiotic division, it forms microspore. Now, later in the mature anther, this is the structure of mature anther. Here, this microspore is formed as pollen grain. Now, these are the pollen grains. When you see the structure of pollen grain, the pollen grain which consists of protoplasm, and a nucleus. So, the protoplasm is present is a protoplasm, the microspore which consists of protoplasm. The protoplasm which is in the pollen grain, the protoplast of the pollen grain consists of dense cytoplasm. There is a dense cytoplasm and prominent nucleus. And this pollen grain is surrounded by, this protoplast is surrounded by wall layers. There are two different wall layers. They are, outer layer is called as XII and the inner layer is called as intine. So, this layer is intine, outermost layer is XII. This is XII, this layer is intine. So, this is the structure of pollen grain. Pollen grain which consists of dense cytoplasm and nucleus. This is cytoplasm, nucleus, surrounded by two wall layers. The outermost wall layer is called as XII and the innermost layer is called as intine. The XII is made up of cellulose, sporopollenin and pollen kit, and the intine is made up of pectin, hemicellulose, and this type of wall material is present in the intine. And this XII is not uniformly distributed. There are certain regions which are not interrupted by sporopollenin. Sporopollenin is not interrupted because the XII wall is made up of sporopollenin and cellulose. Certain regions of this XII, the sporopollenin is lack or absent. These regions are globular in nature and they are called as germ pores. Germ pore. Through this germ pore, the pollen tube develops. So, certain regions of this XII, there are no sporopollenin, they are called as germ pore. Through this germ pore, pollen tube germinates. So, it helps for the germination of pollen tube. And most of the species, this XII is not in the same, it is different species. Depend upon the species, the structure of XII, the wall layers may vary. It is interrupted with different shapes and sizes. the size and shape of this wall layer is varies. Now let's learn the development of male gametophyte. Male gametophyte. The first cell of male gametophyte is called as microspore. Microspore is the first cell of the male gametophyte. So, in the male gametophyte, microspore is haploid. You know, already you learned that microspore is derived from the microspore mother cell. Mother cell. This microspore mother cell undergo meiotic division and it produces microspore. Now this microspore is the first cell of the male gametophyte. Now this microspore, which consists of nucleus and dense cytoplasm, because later when this anther is mature, it becomes the pollen grain. This is the structure of pollen grain, which consists of outer layer, wall layer, dense cytoplasm as well as the prominent nucleus. Now this nucleus of this pollen grain which starts to divide. When it divides the cell into two. So when the cell divides, this is XII, outermost layer, and intine, this is the nucleus and the dense cytoplasm. This nucleus of this pollen grain divides. When the cell divides, it produces wall layers and it forms two unequal parts. Surrounded by this nucleus, there are two unequal parts. The larger region which forms as a vegetative cell and this forms as a generative cell. There are two cells are derived from the nucleus. When the nucleus divides, it is surrounded by a wall layer. The wall layer divides into two parts, two unequal parts. The large part, which is called vegetative cell, and this part, the small part, is called as generative cell. Now this generative cell, which is surrounded by a wall layer. Again, now this microspore is two-celled stage. It is two-celled stage. The pollen grain is two-celled stage. Earlier form is microspore, later form is pollen grain. Pollen grain is two-celled stage. Most of the angiosperms, the pollen grain, it liberate in the two-celled stage. How it is liberated? When you look at the diagram again, here at the junction of each lobe, there are 4 lobes are present, and the junction of each lobe, there is a region which is not interrupted with this endothecium. Now there is a structure called stomium. There is a region, the junction of the lobes, which contains a typical structure that's called stomium. So this stomium and the hygroscopic nature of endothecium helps the dispersal of pollen grain. So the pollen grains are dispersed through this stomium. That's the function of stomium. Stomium helps this anther to disperse the pollen grain when it is mature. So this is the structure, mature anther. At the time of maturity, it becomes the pollen grains. The microspore becomes pollen grain, which is liberated either in the two-celled stage or the next stage, that is called three-celled stage. What's this three-celled stage? In the two-celled stage of the microspore or the pollen grain, there are vegetative cell and a generative cell. Now this generative cell, this is two-cell stage, either the vegetative cell and the generative cell. If vegetative and the generative cell is present in the pollen grain is named as two-celled stage. Next division takes place in the generative cell. The generative cell again divides and it becomes the pollen tube nucleus. Here it produces a tube nucleus and two male gamete. It gives a two male gametes. Let's see the diagram. Here there is intine and this is XII. Here is the nucleus with dense cytoplasm. Now the vegetative cell and the generative cell. Here there is a vegetative cell and generative cell derives two-celled stage. Next stage, this generative cell again divides and it forms the generative cell divides and it develops two male gametes. These are the two male gametes and there is a tube nucleus. This is tube nucleus. These two are the male gamete. This stage is called as three-celled stage. So, two-cell stage which consists of vegetative cell and a generative cell. Three-cell stage which consists of the division of generative cell. Generative cell again divides and it produces two male gamete, that is the sperm, which develops along with the tube nucleus. Now, either this pollen grain disperse in this three-cell stage or in the two-celled stage. If it is derived as if it is liberated in the two-celled stage, it reaches the stigma in the gynoecium. Structure of gynoecium, there are stigma style ovary. In the stigmatic region, when the pollen grain, suitable stigma, when it is falls down, it gets moisture and it starts to germinate. The pollen tube starts to germinate. And when the tube nucleus, when the pollen tube develops or when the pollen start to germinate, it produces, this generative cell divides and it produces two male gamete and the tube nucleus. So, now it is called as three-celled stage. 60% of angiosperms, angiosperms are the normal flowering plants what is around us. So, these plants 60% are liberated the pollen grains at two-celled stage. Few plants, which disperse the pollen grain in the form of three-celled stage. So, today you learned detailed about two questions. One is TS of mature anther, another one male gametophyte. In the TS of mature anther, we give importance to wall layers. There are four different wall layers, namely epidermis, endothecium, middle layer and tapetum. And the structure and function, this diagram is very important. Draw this diagram in the class note and what are the functions of each layer and the structure of the layer. Learn this question. Then male gametophyte, how the pollen tube developed and how the male gametophyte is formed. So, under sexual reproduction, the first part is part of the flower. The essential word is andrecium. Now, in this class, we are conclude the structure of anther. Next class, we are going to continue the structure of gynoecium and its function and its steps involved in the sexual reproduction. Before the next class, you learn this all questions and draw the diagram. Thank you.